Assalamu alaikum students. Today we will discuss viruses. This is the topic from 11th class biology, chapter number 5, variety of life. In this topic we will discuss about introduction of viruses, its discovery, structure, composition and characteristics. Introduction. In early days, that is at the time of Louis Pasteur and Robert Koch, it was considered that virus is a poison and it is associated with disease and death. But the present notion of virus is entirely different. Now we know that virus is consists of nucleic acid and covered with a protein coat, that it is a structure composed of two things, nucleic acid and protein coat. It causes disease. It usually causes disease when it replicates in living cells. It causes disease like influenza, hepatitis, smallpox, and AIDS. Bacterial virus is also known as bacteriophage. Virology, the branch which deal with the study of viruses, is called virology. Meaning, word virus is derived from Latin word venom, which means poisonous fluid. Proper definition of virus is non-cellular infectious entity. This is non-cellular because it is not composed of a complete cell. Non-cellular infectious, it can cause disease and that disease can spread from one person to another person. And it contains RNA or DNA. Inside it, RNA or DNA is present and it is covered by proteinaceous coat and it reproduces in living cells. It can be defined as non-cellular infectious entity which contain either RNA or DNA, normally encased in proteinaceous coat and reproduce only in living cells. Viruses utilize the biosynthetic machinery of the host for its own synthesis and then transfer themselves efficiently to other cell. They are non-living outside any body. They can't reproduce by their own. So we can say they are non-living, but they can reproduce in any living body or use its biosynthetic machinery and then they can transfer themselves from one host to another host. In this way they spread disease. They are non-living. Discovery. First portion has been covered in which we have discussed about introduction of virus. Virus were considered as a poison, the present notion of virus, and proper definition, meaning, virology. The first portion has been covered. The next portion is discovery. The first name is Edward Jenner. In 1796, Edward Jenner first vaccinated an 8-year-old boy with material removed from cowpox lesion on the hand of milkmaid. After 6 weeks, the boy was inoculated with pus from a smallpox victim, but he did not develop the disease. Edward Jenner was a physician in 1796. What he did? He did an experiment. In that experiment, he took material from lions, wounds, and these wounds were present on the hands of milkmaid. And milkmaid has these lions from her cow because animal also has pox and at that time that is called cowpox, and from that lions he took material and injected into an 8-year-old boy. After 6 weeks, almost 1.5 months, the boy was inoculated with pus from a smallpox victim. He inoculated or he injected smallpox virus or take it from smallpox victim but he did not develop the disease. But that boy doesn't show any kind of disease. So he came to know that if he used this method on other persons, he can stop the spreading of that disease which was spreading rapidly, that was smallpox. At that time smallpox was spreading rapidly. So Edward Jenner by an experiment find the way to stop that disease by a method. Later that method was called vaccination. Later, Jenner used material for vaccination from cowpox lions and successfully vaccinated 23 persons. Why the term vaccination was used? Why it is called vaccination? Because the material was obtained from cow. In Latin language, cow is called vacca. Later, the term vaccination was used by Louis Pasteur. This method was discovered by Edward Jenner, but that method was named by Louis Pasteur as vaccination in honor of Edward Jenner because he used that material from the lesions of cowpox. The next name is Charles Chamberlain. In 1884, one of the Pasteur's associate, Charles Chamberlain, found that bacteria cannot pass through porcelain filters, while agent responsible for rabies can pass through these filters. The next name in discovery is Charles Chamberlain. The first name was Edward Jenner, and the name of that process was given by Louis Pasteur. And the next name is Charles Chamberlain. Charles Chamberlain came to know that bacteria can't pass through any filter, or we can say any filter paper, any tissue paper, any porcelain material, any that material which have pore. Bacteria can't pass, but the agent responsible for rabies can pass through these filters. It means some other thing is also present which is responsible for spreading of disease, and that is not bacteria because bacteria is filterable. And he studied rabies. What is rabies? Rabies is a disease which is transferred to a human by bite of a rabid dog, fox, cat, bat, and other animal. That disease is transferred from the bite of rabid dog, cat, bat, and other animal into human, and that disease is called rabies. He studied that disease, and he came to know that the agent responsible for rabies is not filterable. It can pass through any porcelain filter. Why they are called viruses? Because in those days it was related to poison. It was considered that is a poison that is spreading the disease and causing death. That why that's why they were called viruses. The next name in discovery is Whiskey. In 1892, Whiskey discovered that the agent which caused tobacco mosaic disease was filterable. Abnovisky came to know that the disease in tobacco plant, that is called tobacco mosaic disease. The agent responsible for that disease is filterable, means that it can pass through porcelain filter. He obtained bacteria-free filtrate from ground up infected plant and placed it on healthy leaf of tobacco. He observed that filtrate produced a disease in healthy plant. After that, presence of similar filter-passing ultramicroscopic agent was seen in the victim of many diseases including foot and mouth disease and yellow fever. What is foot and mouth disease? Here I am sharing a brief introduction of foot and mouth diseases, but that is not included in your syllabus. Yellow fever. Here is a brief introduction of yellow fever. Okay. Discovery Stanley. The filterable agent were first purified in 1935 when Stanley was successful in crystallizing the tobacco mosaic virus. Chemical analysis of these particles show that they contain only nucleic acid, means DNA or RNA, and protected by a protein coat. This suggested that unlike other form, viruses are of simple chemical composition. With that, discovery portion has been completed. Now we are moving to next portion, that are characteristics. The first character is size. Viruses are extremely small. Their size is very small. Infectious agent. They can cause disease, and that disease can spread from one person to another person, which can only be seen under an electron microscope. They are very small in size. They are infectious and can be seen under electron microscope. Their size 250 nanometer to 20 nanometer. 250 nanometer is present in pox virus and 20 nanometer parvovirus. This is also MCQ. The first characteristic is size. The next characteristic, filterable. They are filterable. In discovery portion, we have read this again and again. They are filterable. They are 10 to 1000 times smaller. It is also an MCQ than most bacteria. So they can pass through the pore of filter from which bacteria cannot pass. It was second character. The third character, they can't grow on artificial media because it is also considered non-living outside any body. They can grow only or reproduce only inside the living organism. We can't grow them in lab. We can't grow them on artificial media like bacteria. We can grow bacteria on artificial media in lab, but we can't grow viruses on artificial media in lab. If we want to grow viruses in lab, we must use our living body or a biosynthetic machinery of that body. The next characteristic is replication. A process by which many copy or replicas of virus are formed is called replication. The process name is replication. Characteristic. They are obligate intracellular parasite. Obligate intracellular parasite. Thus, the viruses are obligate intracellular parasite. Viruses like metabolic machinery for the senses of their own nucleic acid and protein. They can't reproduce their own copies because they don't have any machinery for it. They depend on the host cell to carry out these vital functions. They depend on the host cell. That's why they are called obligate, and they are performing all their reproduction function inside the cell. That is called intracellular. It is obligate intracellular parasite because it can't reproduce or replicate or produce multiple copies of itself without any living organism or without any body or without inside of any living body. This is also very important character, and this is also short question. Disease causing. They are disease causing because they are using living organism or using its biosynthetic machinery. So they, in this process, they are causing disease or they are spreading disease. During reproduction in the host cell, viruses may cause disease. All viruses are generally resistant to broad range of available antibiotics such as penicillin, streptomycin, and other. This is also a short question. Now moving to next portion, that is structure. All viruses consist of two major parts. Inside the virus, nucleic acid is present, that is called DNA or RNA, and a specific term is used for nucleic acid, that is called genome, and it is covered a protein coat that is called capsid. In structure, these two terms are very important, genome and capsid, and these are also separate short questions. The complete, mature, and infectious particle is known as virion. Virions are composed of a central core of nucleic acid, either DNA or RNA, which is also known as the genome and is surrounded by a protein called the capsid. Here you can see genome and capsid are shown. Capsid give definite shape to virion. Capsid is made up of protein subunit known as capsomere. Capsid is also made up of protein and it is further made up of subunits, protein subunits known as capsomere. And number of capsomere is a characteristic of each virus. If we want to identify virus, we have to came to know how many capsomeres are present in its coat. The number of capsomere is characteristic of a particular virus. For example, 162 capsomere are present in the capsid of herpes virus and 252 in the capsid of adenovirus, which cause some common cold. This is also a short question. Each and every heading is also a short question. Structure: nucleocapsid. In some animal viruses, the nucleocapsid, nucleic acid and capsid combinedly called nucleocapsid, and it is covered by another membrane, and that membrane is derived from host cell, and that membrane is called envelope. F, nucleocapsid. Nucleocapsid means genome and capsid combinedly are called nucleocapsid, are covered by another membrane, and that membrane is derived from host cell, then it is called envelope. Non-enveloped viruses are known as naked virions. Morphology, this is the last part of structure. Morphology, animal and plant viruses. Morphology mean appearance, shape, structure, apparently how they look like. Animal and plant viruses may be polyhedron. Polyhedron mean having many. Poly mean many. Hedron means sides. Helical, helical means spring-like. Spiral, enveloped or complex. Animal and plant viruses may be polyhedron, having many sides. Helical means spiral or spring-like. Enveloped or complex. Enveloped, they must have envelope. Envelope, the covering on them derived from host cell is called envelope. Bacteriophage is a virus occur in two structural forms having cubical or helical symmetry. In general appearance, cubical phage are regular, solid, or isohedral, having 20 faces, and helical phage are rod shaped. Here you can see bacteriophage. Many phage consist of head and tail. Here you can see clearly head and tail is present. In those cases, head are polyhedral. The head is polyhedral having many sides and tail is rod shaped. The both shapes are clear in this diagram. Bacteriophage consists of head and tail. Head is polyhedral and tail is rod shaped. Here you can see different types: vaccinia virus, paramyxovirus, herpesvirus, virus. These are different morphological forms or types of viruses. And then there is a tip for you. This topic seems difficult, but it is very easy to memorize. Here I am going to share tip for you. We can divide that topic into 5 parts. The first portion is introduction. In introduction, if we learn about headings or if we write headings on a page and then memorize that topic, it will be very easy. Like in introduction portion, we read that it is it was considered as a poison. The study of virus is called virology. Its meaning, it is derived from Latin word. It is disease causing. Its proper definition, and it is non-living. And in next portion was discovery portion. In discovery portion, we read the first role was Edward Jenner, then Charles Chamberlain, then Whiskey, and at the end Stanley. Characteristic portion. In characteristic portion, we read about its size. It is filterable. It can't grow on artificial media. It is disease causing and it is obligate intracellular parasite. So we can memorize each and every portion separately. Structure. In structure portion, we discuss about genome, capsid, capsomere, nucleocapsid, enveloped, naked, morphology. Here you can see a sequence is present, genome. The inside DNA or RNA is called genome. The outermost covering is called capsid. The capsid, the subunit of capsid is called capsomere. The genome and capsid is called nucleocapsid. Covering outside the nucleocapsid is called enveloped. And the viruses without envelope are called naked virion. And at the end, morphology. In morphology topic, we discussed just that bacteriophage consists of two forms, cubical or helical, and we seen a picture in which bacteriophage was present. It has head and tail portion. Head was having many sides and tail was root shape. With that, we have completed our topic. Thank you for watching.